All right, good evening everyone, and thank you all very much for attending the first Research Tuesdays for 2022, which is called COVID Pawsitive. My name is Kalia Primer and I'm a PhD candidate here with the Adelaide Medical School based at the University of Adelaide, and I'm also your MC for tonight. I would like to start by acknowledging the Kaurna people, who are the original custodians of the Adelaide Plains and the land on which the University of Adelaide's campuses at North Terrace, Waite and Roseworthy are built. Tonight's special presentation demonstrates a really innovative way in which the University of Adelaide is helping with the state's COVID response. Our presenter is Dr. Annelise Chabert, who is a senior lecturer in the University's School of Animal and Veterinary Science. Dr. Chabert has over 10 years' experience as a field epidemiologist and is a One Health expert. One Health is a collaborative research approach that recognises the interconnection between people and animals and plants and their shared environment. It's about striving for optimal health for all species. Dr. Chabert has conducted research on the transmission of diseases including foot and mouth disease, Q fever and MERS coronavirus at the wildlife-livestock-human interface. Her experience in this area has made her the ideal person to tackle the task of COVID sniffer dogs. So tonight you're going to hear about how she discovered that these dogs could be used to detect COVID, as well as the training that's involved in preparing these dogs for service. And as you can see down the front, we've got a special guest. She has brought one of these dogs with her tonight. So following Annelise's presentation, I will come back up and rejoin her on the stage, and we'll conduct our normal Q&A session with you, our audience, both in person and online. I encourage our viewers online, please do submit your questions during our presentation via the Q&A section that should be down the bottom of your screen. But that's enough from me, let's get underway. Will you all please join me in welcoming Dr. Annelise Chabert.
Thank you very much all for coming, and I'm very pleased to see that there are so many people that are actually interested in COVID sniffer dogs. Let's ask ourselves a few questions. Why do we actually need to detect diseases? On an individual point of view, we want to offer people to have access to the best care in the most timely manner. But it is also important to look at the population level. So at the population level, we don't want to have positive cases infecting others. So post-detection isolation is very important to flatten the epidemic curve. But to do so, we require fast and reliable screening tool. What do we have in hands? We've got two kinds of screening tools that we are currently using. One is actually the PCR, which stands for polymerase chain reaction. The PCR is a molecular test that is looking for genetic material from the virus, and in this case, it's RNA. So when you get your swab taken, we actually try to get a bit of the virus on the swab, and then the machine is going to analyze if they can find genetic material from the virus. This is the first one. The second one is the antigenic test. So the antigenic test can detect the presence of specific proteins of the virus. Our problem is that PCR are very reliable, but they are not rapid. We don't get the results fast enough. Rat tests are obviously rapid, but they're not very reliable. And we realize that since the very beginning of this pandemic crisis, what we need is a fast and reliable screening tool. Why are we going to discuss smell? Because I actually think that smell could be this tool. Since 1889, we have had several anecdotal evidence that dogs can detect melanoma and other cancers. We've got the stories from 1889, where a patient actually presented to the hospital because a dog kept sniffing a lesion, and even though she was wearing clothes, the dogs kept coming back at it. So she decided to consult, and they diagnosed a melanoma. And fortunately for her, it was still thin and curable. So actually, she got saved by her dog. In 2013, we had another case, which is presented there, where someone, 75-year-old man, came to a clinic saying, oh, my dog kept licking something behind my ear. And again, it was a melanoma that was detected by a dog that was not trained to detect cancer. So obviously, the dog seemed to be attracted to something and keep licking or sniffing it. So canine olfactory detection of human malignancy is now very well documented. And advanced olfaction is hypothesized to explain canine detection of bladder, breast, ovarian, lung, and prostate and skin cancers. Is it new? Not really. Hippocrates, a Greek physician, which is often considered to be the father of medicine, used the art of smell to diagnose diseases around 400 before Christ. And he actually was teaching his students how to sniff their patient breath and the aroma of disease. His students were encouraged to smell the sputum, blood clots, ear discharge, urine, excrement, and vomit to aid diagnosis of disease. Medical practitioners seem to have put those practices in the back burner, and I can understand why. But other animals have recently been trained to detect diseases. Mice, honeybees, and ferrets also possess the ability to sniff out disease. As an example, United States Department of Agriculture use mice, dogs, and ferrets to detect avian influenza in feces of infected flocks in the US. Rats are also being used to smell out tuberculosis in human samples in Africa. And new research shows that rats can detect up to 70% more cases of tuberculosis in children than standard smear tests. It's quite important. Some insects, such as bees, have also a very acute sense of smell. They can locate the faintest odors, only a few molecules in a room, and they've been shown to effectively diagnose diseases like tuberculosis, diabetes, some type of cancer, and also recently COVID-19. Shall we really be surprised that animals can detect diseases? And I'm going to go back to actually wolves and dogs. So if we take a step back, wolves are the ancestor of dogs, and they are using their olfactory abilities for hunting and tracking animals. So not only stressed animals release alarm odors, which are such as corticosteroid or epinephrine, which can be utilized by predators to locate their prey, but it's also widely known that predators will target prey that are sick. It can be because the prey that are sick are a bit slower, so easier to catch, but it can also be because actually they emit a special odor that helps them to be targeted, well, helps the predator to target them. Just to give you an example, when comparing grouse that were killed by predators to those killed by humans, the one killed by predators had a significant higher worm burden than the one killed by human. Like if the fact that they were actually carrying worm was giving a signal to the predator to hunt them. Some non-canine examples include weasels that actually assess the mice quality before hunting it. Snakes use olfaction to assess which prey are energetically easier to hunt. So they've got a history actually of using their incredible olfactory ability. If we look at wolves or dog, they've got an anatomy and olfactory system and a physiological system that makes them very well equipped to detect and process scent. They breathe and sniff, but breathing is an implicit, effortless, non-cognitive process, while sniffing is actually an effortful and cognitive behavior. They have the ability to smell separately in each nostril. They actually smell in stereo. And that's very important for them because it helps them to determine the source or the direction of the smell source. As air enters in the nose, or as air enters the nose, a small fold of tissue divided into two separate folds, one for breathing and one for olfaction. So as you can see on this diagram, the one for breathing is highlighted in blue, and the one for olfaction is highlighted in red. So the air is actually separated in two folds. The one that enters the region that is dedicated to olfaction, that you can also see on the left, which is the green-brown area, which is very large. So the air that enters this region is actually entering a region that is covered by olfactory epithelium, with highly specialized olfactory receptors. Just to give you an idea, dogs have got around 300 million olfactory receptors, while us as humans, we've got no more than 5 to 6 million. That incredible nasal architecture is supported by a huge olfactory bulb in their brain, which is approximately 40 times larger in dogs than in humans. And that obviously helps them to distinguish and remember a huge variety of specific scents. So canine olfactory detection threshold is 10,000 to 100,000-fold lower than humans. And that's very much the reason why we are already using dogs in Australia and everywhere in the world. They are currently being used to search illegal substances at the border, such as narcotics, weapons, drugs, currency, and also prohibited food and animal products to protect us and our environment and our economy. They are also used to save us during fire research and rescue operation. They are used to protect Australian wildlife from foxes and other pests. We've got conservation program using dogs to actually find penguins, turtles, and other rare and endangered birds and mammals. We've got dogs used by Department of Biosecurity to detect red fire ants, termites, and other pests. But could they be used to actually help us during this sanitary crisis? Could we actually, would dog be the fast and reliable screening tool that we really need? So my aim was to find this tool and to develop it in Australia and to be able to offer that to our communities. But before embarking in such a project, I contacted a French colleague, Professor Dominique Grandjean, who works on canine olfaction. And he shared his first result with me on Facebook, because we are also using Facebook as scientists. And he confirmed that dogs could differentiate between COVID positive and negative samples. So with a fair share of determination and enthusiasm, we decided to investigate dogs' accuracy and fitness for purpose in Australia. So I contacted other agencies, including the Australian Border Force and the Department of Biosecurity, to know if they would be willing to engage in such a scientific adventure, and they confirmed. That's the reason why we actually managed to do this project on a very global level. Before asking a dog to sniff out for COVID-19, we actually have to teach him what he has to search and how to search. We actually started the training of the COVID-19 medical detector dog in Adelaide, so at the University of Adelaide and the Roseworthy campus, and also in Melbourne in collaboration with ABF. The training is based around the key principle of creating a positive and a rewarding association between a smell, which is a target odor, and the first smell that we actually use is Kong. So we brought a little piece of Kong. Kong is a little piece of plastic. All our dogs are actually initially trained to find this odor. So if you've got Kong or yourself, they will get very excited and try to find you. And then, when they manage to find the odor, what they have to give is a conditioned response. For us, it's a sit and stare. So you've got lines of different samples with different odor, and when the animal finds the right odor, he will sit and stare, saying, I found it. And that when we do, or what Alex does, because Alex is our chief dog trainer, the dog will be rewarded either with a toy or with food. We've got some dogs that prefer toys and some dogs that prefer food, and we give them what they prefer. Quake, for instance, prefer food. When they've got the training system or the training process well established, then we move to a new odor. And the new odor for us was the VOC linked to the infection with COVID-19. It sounds easy to say that, but to actually train a dog to detect people who are infected by COVID-19, we needed to provide them with a lot of samples from different people that were infected by COVID-19. A sample is something quite easy to get. We were using gauze, putting gauze under the armpit, wait for 2 minutes or longer if you've got more times, and when you finish collecting the sample, you put it in a little plastic, and that was coming to our lab. That's what I'm going to show you here. So normally we were using gloves when handling the samples, but that was done for TV, so we were a bit less strict. So we actually had to imprint them with 80 positive samples from different people, because we don't want them to find one person, we want them to find COVID. And once that's done, when they've been imprinted, then they've got this exercise that we've just seen, where the dogs have to go through each hide and say, negative, 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 positive, I sit, I stare, give me my reward. Why did we use sweat? Do you have any idea of why did we use sweat? No? Am I allowed to ask questions to the public? No? We use sweat for 2 reasons. First, because we are always sweating. So it's a very good and a good thing to use in operation. When I say we are always sweating, it's true. We're actually producing 1,450 liters of sweat per year. So we've got sweat. It's very easy to use in operation. The second reason why we use sweat is because sweat is a non-infectious material. We know that because we actually did PCR, so you know, the method polymerase chain reaction, trying to find the virus in the sweat of people, and it always came back negative. So we know actually that sweat is a safe sample to handle. Safe for the people who were handling it, and safe for the dog. Our problem at the beginning of the program is that, which is good for Australia, is we didn't have any cases in Australia. We knew it was coming, it was all over the world, but we had a very strict 0 case of COVID policy, which was very good. And so we couldn't actually rely on getting 80 sweat samples from people around Australia, because there was no one having COVID-19. So we heavily relied on our international partners. France, the UAE, and also Belgium provided some samples. And we actually shipped samples, hundreds, thousands of samples from them to train our dogs. We've then, once our dogs were trained and we were confident they actually were detecting COVID-19, the piece of the puzzle that was missing was how accurate they are. That's the question that every journalist will ask me. How accurate are your dogs? So we embarked in a very tedious exercise, which was to assess the accuracy. we've completed 1,333 runs. So a run is what you are seeing now, where the dog has to find the positive sample. We've done that with 1,203 positive cases. So everything was double-blinded, so meaning the person that was actually handling the dog didn't know where the positive sample was, and the person that was recording the data didn't know where the positive sample was. And then we were comparing what did the dog say to the PCR results. I just want to highlight that all the samples we were getting them from people that were symptomatic or asymptomatic. Actually, that didn't really matter. We had two groups of samples. And we were getting, but we knew that we were getting the samples from people that were in the first week of the infection. So we knew that they were truly positive cases. The results were very impressive. Our sensitivity, which means it's when you look at the sensitivity of a test, is how often the test is going to give you a positive result when you are truly positive. So sensitivity of the dogs was ranging from 87.6% to 100%, depending on the dogs. And the specificity, which is the ability of the test to give you a negative result when the sample is truly negative, was ranging from 96% to 100%, so very, very good too. The dogs that were very sensitive were a bit less specific, and the dogs that were very specific were not very sensitive. So what you gain, you lose it somewhere else. So that was actually very comforting. We were like, we've run more than 1,000 runs, and our dogs are doing very well. We were ready for our operational trial, the first operational trial, which happened in Sydney Airport. And what you're going to see is actually when it's not the true trial, is when we actually want to train the dog to actually work in an environment where it's going to see a lot of people. We kind of try to make noises to see if the dog is going to be disturbed by the noises or not. The truth is that the Sydney trial didn't go according to plan. We had a very competent dog in the control setting, and we brought them to the airport, and the dogs didn't find the first positive PCR person. That was shocking to me, because we never had a case where all of our 15 dogs, we trained 15 dogs, where all of the 15 dogs miss a sample. It can happen that sometimes we had one dog missing a sample, but it never happened that we had all the dogs missing one sample. So that actually was putting a bit of stress on my shoulder, and I was like, what is happening? Second sample, second person coming positive, again, all the dogs are missing it. After a few sleepless nights, I started to build a hypothesis of why are the dogs missing it. And I'm sorry, I'm going to try to not be too technical, but it's very important to understand what's going to happen now. So we were getting samples to train our dogs under this curve, the red curve, when people are truly very infectious. The nasopharyngeal swab that we are taking, and where you can still have a positive PCR result, is the blue line. You can have a PCR positive result for a bit longer, because the PCR can actually detect remnant particle of the virus. So the virus is not live anymore, but you've got a bit of genetic material of the virus that is still present in your throat. Very often, when that's the case, the cycle threshold of the machine that is doing the PCR is very high, meaning that the machine has to run many cycles to actually find the piece of the virus. When we were training our dogs, we knew that we were there. When we are taking a sample from someone at the airport, we don't know where they are. They actually were supposed to be PCR negative while boarding the flight. So it was very surprising that they were PCR positive upon arrival. So the reason why they were PCR positive upon arrival is because each lab got different cycle threshold reference. And so it's possible, and we never managed to actually get the cycle threshold of those people. It's possible that the people that all our dogs missed were there, meaning they are still positive, but they are not anymore in the infectious period. That was my hypothesis when I was trying to understand what was happening in Sydney. It was a very long 3 weeks where we were trying to understand what was happening there. So what we've done is we started to screen a lot more samples in control setting, but also in Adelaide Airport, where we knew if the people were from the first week of infection or not. We knew exactly where they were at. And we managed to confirm that it was actually true. The dog can detect people there, but they cannot detect people that are not infectious anymore, which is actually very good, because that means that we've got dogs that can detect people only when they're infectious. And we also realize that the dog can detect people a day before the PCR, because it takes a bit of time for the PCR to come up positive while the cells are already infected. So after we realized that, we were much more confident to do a trial in Adelaide Airport. And that worked very well. We knew the strengths and the limitation of this test, and we realized that the dog were only going to detect people in the infectious period, and we actually worked with assay pathology. For the people that were negative or positive, we knew exactly where they were at in their cycle. So it was a success, but we still had 2 issues that needed to be sorted out. The first one, at this stage, we were still relying on those sweat samples. So nurses from assay pathology were collecting samples from people that were just landing. The good thing is that when you spend 12 hours on a flight, you are sweaty. So we had very good quality samples. The bad thing is that it was taking a lot of time. So it was not sustainable to actually screen hundreds of passengers. The second problem was that until now, we trained our dog using sweat samples from people who were infected. That is good for a trial at the university, but our aim is to actually be able to train as many dogs as needed. And we cannot rely, or it's logistically impossible to only rely on positive sweat sample to train dogs. So we worked on actually developing what we called a training aid. Oops. A training aid is a surrogate scent of COVID-19. This is only taking 5 seconds in my presentation, but actually it took us months to actually develop the right training aid. So it's something that is not infectious, and I can tell you it's a cocktail of protein that we are using, which are not infectious. And when the dogs have been imprinted on this training aid, then they can sense and detect people who've got COVID-19. We are putting this training aid on different media. We are putting on masks, on gauze, and on socks. We confirmed that the dogs that were trained with this training aid were able to actually generalize and find people that were positive with COVID-19. So that's the first thing. We managed to find a way to actually upscale this capability. The second thing was to actually make sure that our tool was fit for purpose. We don't want to rely on sweat sample anymore. We want to screen people directly. And that's where we are at. We are currently putting the training aid on socks of people. Everyone was negative here on the video that you just saw, and the dog will sit in front of the person that is wearing the sample with the training aid. Our aim is to get to this path. This is Quake screening people, and he will sit in front of the one with the target odor. That will be amazing if the dog can do that, not only at the airport, but also in hospitals. So where are we at now? We are at the point where we are collaborating with SA Health and SA Pathology to deploy dogs in health settings. We won't have problem to train more dogs. We think they actually will be able to detect people in health settings. But we might actually struggle to find enough dogs and enough dog handlers. So our step is now to train nurses from SA pathology who will be able to operate the dogs, so they don't rely on us to actually operate the dogs in those kind of settings. Canine detection of diseases is actually a little or maybe a big revolution. It's a new diagnostic and screening tool that is not based on pathogen or antigenic search, but is based on finding volatile organic compound. It's opening a totally new area of research. There are still many unknown, and we've still got much more work to do, but we've proven that it's actually useful for COVID-19. It might be very useful for other diseases that can obviously have an impact on our health, but also on national security. So it's getting very popular to discuss about this recent scientific research. And I think the potential applications are endless. Some people are telling me that now that we will have to live with COVID, maybe we won't need to actually screen people anymore. It's true, we will have to live with COVID, but not everyone will be able to live with COVID, and I think we will still use screening tools in places like oncology departments or for people that are actually receiving transplants because they are immunocompromised, and we will have to make sure that we don't actually bring any other diseases. This project is not only showing that dogs are able to do it, but it's also showing that a lot of different agencies are able to work together nationally and internationally. I'm not speaking about everyone during my acknowledgement slide, which is actually lasting for 2 minutes, because we had more than 40 entities involved in the project, nationally and internationally, at federal level and at state level. So it is, I think, a wide success to be able to actually do this kind of transdisciplinary project involving so many agencies. And I'm reporting to the World Health Organization. Nothing would have been able without the incredible team that we've got in Adelaide Uni, and also without the support of Adelaide Uni, who also finance part of the project. So we collaborated with a lot of countries, actually 22 countries, and we share all of our data in real time to anyone that might need them. I'm now ready to open the floor for questions, and I hope you enjoy this talk. Thank you very much.
Thank you so much, Annelise. That was so fascinating. I love how you can tell when you play those videos that those dogs are so excited to be out doing their detective work. Let me just put my laptop up here. All right, we do now have some time for questions. I've got the online questions, so for everyone watching online, please submit some questions if you've got any. We do also have Liz running around with a microphone, but I might kick us off with one that we had that was pre-submitted, which is how long does it take to train these dogs to go through that whole process?
It depends on the dog, and it depends on if they had any prior training. So, for instance, if they know how to sit and stare in front of the target odor. But a rough number will be 8 weeks.
Yeah, okay, well, it's
from scratch.
It's a fair while, yeah. But that's not too bad. So another question, and this is sort of tied into one that we've just gotten online as well. Are particular breeds of dogs better than others? And Tina online has asked, have you trained any other breeds of dogs, for example, beagles?
So we could train many different breeds, and I think beagles will be a good breed. We are mainly training Labradors, but many breeds could do it. But what we need is obviously not only a good nose, but the dogs need to be driven and be excited about their work. And so we need dogs that are driven and that got a good work ethics. And that's the reason why we work mainly with Labradors. Another thing is that those dogs are going to be in contact with the public, and we want the public to have a good perception of the dog, and Labradors are also good in this sense.
Of course, yeah. And you can tell they love their job.
Yeah.
Do we have a question from the audience? Please go ahead.
Thank you, Professor Chabert, for your fantastic presentation. I wanted to ask about the volatile organic compounds and how have you been able to try and work out what it is in those volatile organic compounds that mean that a case is positive and that the dog sniffs that? Is there some marker? Are we able to describe that?
This is a very good question, but I'm sorry, I won't be able to share everything that we know on this topic. But yes, there are viral proteins that are being used to detect who is positive and who is negative. And we are currently working on really pinpointing which protein it is. We are currently using a cocktail of protein, and I will leave it there.
That's good. That was going to be one of my questions if someone else didn't ask it, but we'll just have to keep an eye out for the answer.
Thank you.
Do we have any other questions from the audience at this stage? Yes, we've got one up there.
Thanks, Annalise. Did you have any indication as to why some dogs were better than others?
No, we don't actually really know why some dogs are better than others. I think it's very often linked to their training history. So the dogs that actually have been trained for other substances and that were really used to this kind of work kind of had better results. But it was actually not significant, not statistically significant between the two groups of dogs. We had a group of green dogs and a group of very well-trained dogs. And it was a bit better on one side, or the trained dogs were a bit better, but not enough to actually say that's the only reason why. We know that dogs, when they get older, they kind of start losing their olfactory capabilities. But on the same toll, they are also getting better at remembering and at processing information. So I cannot really give you an answer of why some dogs were better than others. I think it's a bit the fact that we had some dogs that were very sensitive but not as specific, and some dogs that were very specific and not as sensitive, is a bit like every test. You cannot be good at both. So I think it's really dependent on their character.
I think that's very interesting. We've got another good question online from Janet. Janet says, do we have one in person? Okay, we'll just do that and then we'll come to this online one.
Thank you for a very interesting presentation. I'm just curious how tolerant are the dogs to multiple olfactory senses? So can they, if they're just trained on COVID-19, are they limited to that, or can they do other senses?
So we've got mainly two big groups of dogs. We had the 15 dogs that were trained at the beginning of the program. Half of those dogs were coming from the Australian Border Force, and they were trained on other substances, and actually we only took the dogs that were trained on explosives because we didn't want to have any other scent interfering with our detection. So explosive and no drug dogs. The dogs that we are currently training now are only trained on COVID-19, and the reason why we only train them on COVID-19 is, again, we don't want to have any interference with other things that they could actually come in contact and give a false result because of something else. But they could. The dogs that are trained from the Department of Biosecurity, Department of Agriculture, are trained on 50 or even more scents. So they can have a good library. The thing maybe I can mention is until the Delta variant came, the dogs could actually detect any variants, and we didn't need to retrain them. Omicron kind of put a little kick in the dance, and we realized that the dog trained for Omicron needed to readjust. The dog trained for Delta needed to readjust for Omicron. So actually now we're training for both variants just to make sure they're not going to miss it. And if we had 10 more variants, we can just add that to the library.
Fantastic. So this is there's no more in person just right now, I'll go to this one from online from Janet. She asks, or says, I do dog agility training and some of my friends do competitive tracking. Will you be interested in working with amateur dog trainers for this? Maybe that could be helpful in terms of scaling up if you want to train nurses as well.
Yes, I'm always open for discussion and new ideas, so I will never say no.
Yeah, fantastic. Do we have any more in-person questions? Any more from the audience? Not right now. All right, so I've got... Oh, there is one down the front. Thank you.
Thank you, Annalise. That was very interesting. When you say you're adding to the library with the different variants, is that adding to the protein cocktail, or is that another detection that you're going to have to...
Yeah, sorry, I was meaning their mental library. I think the dog's got a library of all the scents that they've been through. And so if we've got new variants that are totally different from the other, they shouldn't, but if they are, we can just add another odor to them, which is mean like a new variant odor to them. It's a bit like when we train dogs to detect some type of meat. If they are a biosecurity dog, you can add many more type of food products. Well, for us, they can be trained for one thing and many other things.
Thank you. Any other questions from the audience? We do still have a couple of pre-submitted ones that we can keep going through if we're happy to keep going. We've also got a few online. So Tina from online has asked, how long can the dogs work? Do their olfactory senses become overwhelmed?
So I don't think the olfactory system can become overwhelmed, but they can get tired like everyone else. Dogs can screen roughly 300 people in less than an hour, but we don't make them work for an hour. What we do is we make them in quite intense fashion for 20 minutes, and then we rotate them. It's just because we obviously love our dogs and we don't want to push them too hard, so welfare of the dogs is something quite important to us, so we rotate them every 20 minutes. They could potentially do more, it's just because we actually don't want to make them do more.
Yeah, they need their breaks too. We have another question. Were there any from the audience? Nope, we have one more from online. Ruben has asked, can a pet dog be trained to detect health conditions of their owner? For example, can a pet dog be trained after an owner's cancer has been removed and trained to detect for potential future metastasis?
It's a very good question, and I feel that the answer is going to be no, because for a dog to be accurate and reliable, it needs to be walked. So if it's only exposed to one person, then it's not really worked. So it's a bit like our dogs. We want to make them work every day except weekend. If they stop working, they might forget what they actually have to do. So it might be a hit and miss for this one. Maybe the dog will say something, or maybe the dogs won't say anything. And if we don't actively walk the dog, I'm not sure what he will do.
That's interesting. Do we have any further questions from the audience? No, we've gone through the main ones that we've received online. Maybe I'll just ask another one. This is a bit of a throwback. Right at the beginning of the pandemic, people were talking about cats catching COVID, dogs catching COVID. What have we figured out about that? And is there any chance that the dogs are at risk of catching COVID?
This is a very good question, and I actually am surprised that no one asked it before. So dogs have got very, very, very low susceptibility for catching COVID. They don't actually have the right receptors, and if they are in a very infectious environment, they might get it, but they actually don't get sick of it, and they don't transmit it. The situation is a bit different with other species, and for instance, ferrets are known to not only catch disease, but can replicate the virus. The virus can mutate and they can transfer it to other ferrets or even to humans. Same thing with hamsters. The situation with mice is also a bit more concerning. So no, dogs have got very low susceptibility, but we couldn't use other species for the work we are doing, exactly for this reason. Another thing that maybe I should mention, at the beginning of a program, there was a lot of hesitancy for two reasons, because first we were importing samples from overseas. That's in a COVID-free country, it was a bit bold. And the second is people where we didn't have any information about how dogs were going to react if they had to be in contact with COVID-19 and will they get sick or not. So we've done a PCR on our dogs every 3 weeks. All of our 15 dogs from the first batch were actually PCR screened every 3 weeks for 2 years. And none of the dogs ever come back positive. And all the dogs that are included in our international program are also PCR tested, and none of the dogs ever come back positive. So I'm in the stage where I'm fairly confident that we are safe.
That's good. Very good to hear. Okay, if there's no further audience questions, I think we might wrap up there. I would like to finish by thanking our speaker tonight, Annelise Chabert. Please join me in thanking her.
Can I add something?
What's that?
Can I add something?
Of course, go ahead.
Sorry, and I just realized that I forgot to add this last slide. We are actually seeking support because we would like to train more dogs, and that's really to be used for the community. And we also would like to start working on other diseases that might have an impact on our communities. So if you feel that you want to donate a bit more than sweat and also some coins, don't hesitate. Thank you.
Thank you very much. And thank you to you, our audience, for coming along tonight, and thank you all so much for your fantastic questions. We look forward to inviting you back next month in April, when we're going to hear from two of the university's newest talents in cancer research. They're going to discuss some new world-leading facilities and capabilities of this new cancer research institute that's being established here, the South Australian Immunogenomics Cancer Institute, better known as SAGENCI. So be sure to sign up for our mailing list to receive the latest information all about that and any other Research Tuesdays news. Thank you all very much for tuning in once again, and we hope to see you next time. Good night.